Okay, hi everyone. So we are pleased to welcome Professor Michael Gromiha from IIT Madras in India and Dr. Somya Krishnan, who is a recent PhD graduate from the Gromiha lab. And they will be talking about a very interesting topic today, which is probing RNA small molecule interactions with machine learning. So over to you, Professor Gromiha.
Thanks, Nivedita, for inviting us to give you a talk at this webinar. It's our great pleasure to share our research results on nucleic acids in our research group. I'm Professor Michael Gromiha from the Department of Biotechnology, IIT Madras. And Dr. Soumya carried out her doctoral thesis work under my guidance in protein bioinformatics lab at IIT Madras. Although we are mainly focusing on proteins, we have made significant contributions on understanding the structure and dynamics of nucleic acids as well. So here we highlight some of the aspects what we have contributed on nucleic acids. In fact, I started the work on nucleic acids maybe 3 decades ago, that is 1994. Okay. Yeah. So here we carried out the analysis to understand the stability of both DNA and RNA. And then using the available 3D structures, we developed some scale for the stiffness or the flexibility for the trinucleotides of DNA and RNA. And we use these parameters to understand DNA binding and how the binding is important for the crow DNA recognition and so on. Then we extended this analysis to understand the factors which influence the recognition of these nucleic acids as well as proteins. We carried out a systematic analysis and multi-canonical Monte Carlo technique for sampling to get the interaction between base amino acid interactions as well as extended to protein nucleic acid interactions. So we dissected the various factors, specifically cation by interactions, hydrogen bonds, electrostatic interactions, as well as disorder to order transitions, to account the residues which are interacting in the surface interface, as well as the binding affinity of these complexes. So we developed various algorithms for predicting the binding affinity of protein nucleic acid complexes and the change in binding affinity upon amino acid mutations. Recently, we related this information to understand the mechanism of the molecular basis of some diseases, specifically cancer, and we tried to predict these cancer-causing mutations in the specific cancer census genes. Then we contributed in last CASP16, right? Samya mainly involved in the calculations, and our group ranked 15th in the DNA RNA monomers category, and we also contributed in the blind prediction of complex water and ion ensembles around RNA in this CASP16, and one of the contributors in the proteins. And recently, once Soumya joined our group, we focused on RNA ligand interactions. So we have focused to develop a database for the experimental binding affinities of RNA ligand interactions, and we use this database to understand and predict the binding affinity of this RNA along with these small molecules. And recently, we developed a novel approach to identify the druggability of these protein, these RNA structures. So now I hand over this session to Dr. Soumya, and she has significantly put all the efforts to the study, and she will give much more flavor about the RNA ligand interactions, right, and how we develop several algorithms as well as applications in RNA-targeted drug discovery. Soumya, please carry on. Thank you.
Thank you, sir. I hope I'm audible.
Yeah, it's fine.
Yes. So thank you for the introduction, sir. So yeah, we'll start by asking the question. So what question we asked was, why should we target RNA why should we consider RNA as a drug target? So there are three primary motivations here that I've shown in the slide. So first, if you look at the pie chart on the left, we see that existing FDA-approved drugs target only like approximately 0.05%, like 700 proteins that are encoded by the human genome. But there is a large fraction of the human genome that encodes for both coding and non-coding RNAs. So the abundance is one factor. Second is they are functionally really versatile. They are involved in almost all major disease pathways. And thirdly, they also have structural complexity similar to proteins. So here are a few examples of disease-associated RNA structures that are shown here. So we have internal ribosomal entry site, which is important for translation in viruses. We have splice regulatory elements, which have been recently used as the drug target for risdiplam to treat spinal muscular atrophy. We have repeat expansion structures like the ones found in Huntington's and myotonic dystrophy, and we have miRNAs, which are involved in several cancers. They are important biomarkers in cancers. So all these structures have ligandable binding pockets that can bind drug-like small molecules. So these are the motivations behind why should we consider RNA as a drug target. But we are not the first people to think of this. There are obviously previous studies on this. There are lots of experimental studies to identify RNA targeting molecules, a few of which I have listed here from this review article. So in this, we can see that there are two fractions of assays, namely hit identification assays and hit validation assays, where a large library of small molecules is experimentally screened against different RNA targets. But you can see that the hit rates are really low. It's a real needle in the haystack problem. And also the affinity or potency of these ligands is mostly in the micromolar range, requiring extensive optimization to turn it into a lead compound. So because of these things, there are also other limitations, like they are sometimes not quantitative. For example, if we consider small molecule microarrays, the data is like qualitative data, whether it binds or whether it doesn't bind, but there is no affinity data involved in that. And then the experimental design can be very specific to the target of interest. For example, TAT transactivation assay is very specific to HIV, HCV replicon assay is specific to hepatitis C, etc. So generalization is also a problem in some of these assays. So in these, due to these, what we asked ourselves was that whether we can develop computational hit screening and prioritization methods, which could be a good alternative for filtering small molecules against RNA structures. So the key lacunae that we wanted to address here was that there were no databases exactly for RNA small molecule interactions and their binding affinity, but there is a lot of data from numerous experimental studies available in literature. So once we create that database, next we wanted to predict binding affinity. So the binding affinity is an important quantifier of the strength between the interaction between the RNA and small molecule. So most of the existing methods are classifiers. A few of those I have listed here. And these methods are also heavily dependent on data augmentation with protein ligand datasets, which is not representative of the RNA binding space because the characteristics of binding sites in RNAs are very different from that of proteins. They are more polar, whereas proteins prefer more of a hydrophobic core pockets, which are non-polar in nature. And there is also no computational method when we started this work for identifying RNA targets in diseases. And there was no basic end-to-end computational pipeline for RNA-targeted drug design. So like I said, we started with creating a database in the first place for RNA small molecule interactions, and this is published in Journal of Molecular Biology. The database is called RSIM, which stands for RNA Small Molecule Interaction Miner. So we curated the data from two major resources. So one is PubMed, and there were two small databases at that time. One is SMMRNA and the other is RBIND. So we collated the data from these databases as well. And we annotated the data extensively with different types of annotations like RNA, small molecule, and interaction annotations. We gave specific emphasis on the structure of the RNA. So we gave predicted secondary structure based on the sequence using VNRNA. And if the experimental structure is available in PDB, we accounted for that. And we also provided predicted or experimental complex structure. And we visualized it in the each entry in the database. So this is the front end of the database. So we have the version 2 is online now, and we are collecting more data to update it regularly. There are different options available for the users to browse through. We have several basic and advanced search options. We have detailed statistics. We have tutorials to help the users get through the data. Users can also both upload and download the data. A lot of options are available to explore. So this is a sample entry from the database. In this entry, we have an entry ID of course, and then we have different panels displaying the information that we've curated and annotated. So we have secondary structure diagrams, the sequence, tertiary structure of the RNA small molecule complex. In this case, it's experimental, but if it is model, the word will be model here, will be mentioned explicitly. We have experimental conditions through which the binding affinity data was curated, and the literature source from where the data was actually collected. So we have, this is a sample entry. We have like 2,624 entries right now from 483 RNA targets, 1,393 small molecules, and the interactions span 27 organisms. So the total data was collected from 227 publications from 1977 to 2025, and of course the 2 existing databases. So KD is the most popular activity type. Some more statistics are given here, like which diseases are common, which experiment types are covered the most, and what are the different RNA subtypes that are covered in the database. We also compare the different features of our database with existing databases. So we have 4 existing databases which are smaller, which at that time were smaller than us. And we have given what are the distinguishing features here. So we have the most number of entries which supports machine learning, because machine learning is heavily dependent on the data. And we also covered the 3 major RNA subtypes, namely coding, non-coding, and synthetic. We also have structure annotations which are lacking in the other databases. So RSAM has been popularly used now since publication. So it has become the dataset of choice for training RNA small molecule binding affinity prediction models. Notably, BOLS2 from MIT, SMRTNet from Tsinghua University, and Deep RSMA from A*STAR, Singapore. They have used our dataset for training their binding affinity models. It has been used for benchmarking purposes to assess predictive models and for docking programs, and to screen RNAs for ribotac discovery. So these are some applications which RSAM has found in literature. So once we created the database, now we wanted to use it to predict the binding affinity between RNA small molecule pairs. So for that, we also wanted to understand what features govern this interaction mostly. There are 2 publications associated with this. One is from Briefings in Bioinformatics in 2024, and we have a short letter to the editor published in 2025. I'll get through that in this. So once we have an RNA and a small molecule pair, what we do is we compute a set of close to 2,000 features. So we have RNA sequence features, we have RNA secondary structure-based features like what we call structure status descriptors, and we have n number of small molecule features computed from the moderate package. So what we do is that we split the data set basically. We took the data that corresponds to only the KD, because the KD was the most abundant affinity type in our database, and we split it into different RNA subtypes. So these are the 6 subtypes that we have: aptamers, mRNAs, repeats, ribosomal RNAs, riboswitches, and viral RNAs. So for each category of RNA subtype, we built a linear regression model to predict the binding affinity given these features. So how we did that is we developed our own forward feature selection workflow. This is patented in India. So we've find the optimal combination of RNA features and small molecule features that can give the best predictive performance. So the metrics are provided here for each RNA subtype. So this workflow can handle multicollinearity as well as pairwise correlations in a dynamic fashion. And we also compared the results with the existing method. So like I said, there were no regressors at that time when we started this work. So one of the regressors that was prominent at that time was this HIV TAR QSAR model. HIV TAR is the transactivation response element. It's like the model RNA in these kinds of studies. So it's for this, the Amanda Hargrove group from Duke University, they have collected 48 different compounds, and for these 48 compounds, they have developed this regression model, QSAR model. So this is their performance metric. So when we initially tested our viral RNA model for this data set, our metrics were not acceptable. But when we actually looked at the data set carefully, we found that there is a dimethyl amyloride scaffold. A few examples are shown here. So this scaffold actually was very dissimilar to our training data set, and that was the reason the model was unable to perform. So once we removed those compounds, the performance was actually good, better than what we expected. So we refined the model further with examples of these dimethyl amyloride scaffold, and finally we could obtain a HIV TAR specific model that had even better predictive capabilities compared to what was reported for the HIV TAR QSAR model. So these are the features that I was talking about that got selected through our forward feature selection workflow. I have split it into two categories for each RNA subtype, namely RNA features and small molecule features. So we have a lot of triplet dot bracket notations. This is called triplet status descriptor. So here the center nucleotide is given on the left, and there are two flanking nucleotides that are part of this triplet. We also have the structure status descriptors where there is a base pair, pair, or there is a base pair and an unpaired base pair. So we have we are looking at frequencies of these interactions across the sequence. So that's what we have found. Since this is cumbersome, I have put it into a set of key observations. So what we found is that when RNAs and small molecules interact, for each RNA, there is a specific KMO composition that is dominant. And there are a few structure status descriptors that are dominant. And these are very specific to the RNA subtype. For example, MIC0. MIC0 stands for mutual information content, which talks about lower complexity in the sequence. And this is more prominent for repeats. And it got selected for the repeats model. The GCA is a structure status descriptor where we have a GC base pair opposite an unpaired adenine and in a bulge region. So these kinds of regions are more prominent in aptamers, and this has been shown in literature to improve binding to the small molecule and stability. We have this unpaired adenine stretches in miRNAs which increase the binding site flexibility, etc. So we found these interesting structure-based descriptors when we built the model. And also we got small molecule features which are dominated by polarizability because of the nature of the binding site, and autocorrelation features related to ionization potential, electrotopological state, etc. So these are the kind of features that we got. We have also developed a web server to host this model. So we have the front end of this web server, we have 6 RNA subtypes from which the users can select. We have provided a box to paste the RNA sequence, and then the user can provide the small molecule in SMILES or in SDF format, and they will get the prediction. So this is the URL for the web server. Please do check it out in your projects. And for batch calculations also, we have provided the source code in the GitHub repository. So RSAPred has also been used really widely since publication. It has been used to compare against newer models that get developed in literature. I have shown here 6 different models that are benchmarked against RSAPred. All of them are deep learning based methods. One of them is sequence based, while other 5 are structure based methods. So here we found that RL affinity, which was published right after ours, actually it had several issues with the data set as well as with the comparison with our method, and we submitted a letter to the editor, which I spoke about when I started this objective. And yeah, when we looked at the comparison made by existing methods, we still find that miRNA and repeat models, these models are not still not improved over by the deep learning methods. So this is one of those cases where machine learning has an advantage over deep learning models just because the data is scarce and machine learning is better suited here. So for 4 out of 6 RNA subtypes, however, we have better models from deep learning methods. This is the current status. So thirdly, like I said, target identification methods are really rare or scarce in the space. So we wanted to build a method that can, given an RNA structure, it can actually predict whether the RNA has a druggable binding site or not. So this was published in Nucleic Acids Research last year. I wanted to give a primer on druggability. So like I said, target identification is the primary step in drug discovery, and there are 2 conditions that a druggable target must satisfy. So one is it should be ligandable. Ligandable meaning there should be a binding pocket that can have a small molecule inside it with high affinity. And the second thing is the binding of that small molecule should lead to potential regulation in downstream disease pathways. And this regulation is what makes it druggable, because it's at that point the small molecule turns into a drug. So that's an important point. These are the existing methods in literature listed here. 90% of them are for proteins, and they can be split broadly into whole target level scoring methods and binding site level scoring methods. For RNA, there are 2 methods available in literature, but both of them are ligandability prediction methods, and there are no standard data sets available for this problem. So we first started with a very careful dataset curation process to build this model. So first we curated the positive data set of RNA small molecule binding sites from PDB. So we finally had 399 RNA small molecule complexes. We omitted certain complexes like synthetic RNAs because they don't have disease association, ribosomes because they're highly promiscuous and they have a lot of binding sites, like 60 to 70 binding sites, and then ion-based riboswitches because ions are technically not small molecules. So for these reasons, we omitted these RNA structures, and we used a binding site cutoff of 6.5 angstrom to extract the residues in the binding cavity. To incorporate flexibility data, because NMR structures are highly flexible, they have a lot of dynamics. So if there are NMR structures available, for each NMR confirmation, we have a separate data point in the data set. And there are also cases like NAD riboswitch or tetrahydrofluoride riboswitch, where there are multiple binding sites present in the same RNA structure. And for each binding site, for the same ligand, there is a difference in the affinity, which says that the binding event is not the same between the first and the second. There is some level of cooperativity or allostery involved there. So we considered these also as individual entries. We resolved the redundancies in the data set through infernal CM scan, and we clustered the data basically. And also some cases where RFAM family annotations were not available, we had to do manual annotations to remove the redundancy. So we finally got 56 positive binding sites. For the negative data set, we used 3 parallel approaches. So we had backbone motif search, flexible blind docking of the ligands that are present in RSIM database, and then we have binding site prediction methods. So we combined all these 3 and removed the redundancy again, and we compared also with a positive data set, and finally we had 92 negative binding sites. So this complete approach also we have patented in India. So this final data set was 148 binding sites, and these binding sites were featureized mathematically using these structure descriptors. So we have 69 descriptors listed in this table, from 7 different feature categories. So we have composition in the binding site. We have pharmacophore elements like hydrogen bond acceptors, donors, polar groups, hydrophobic groups, etc. And then we have surface area descriptors. We have principal moments of inertia descriptor, which talk about the shape of the binding cavity. And then the roughness descriptors, we have torsion descriptors, backbone, pseudo and sugar torsions. And we have nucleotide type and sugar puckering kind of descriptors. So these descriptors were used to featureize the RNA binding site. And then we built classifiers basically. So given a binding site, whether it is druggable or not, and the probability that comes out of the classifier becomes kind of like a score to say whether that how highly druggable is that binding pocket relative to some others. So we built different classifiers basically. And here in this table, I have given the results. The test metrics actually come from a special external test data set, which we call as the blind test data set. So far, what is special about this is, so far what we have trained with is hollow structures. So they're already bound. The RNA is already bound to the small molecule in PDB. But there are cases where apo and hollow structures will have wide variation in the structure. So we curated a specific data set of empirical and modeled apo structures with verified druggability specific to SARS-CoV-2 genomic elements and human MIR21 onco-mRNA. So we combined these and formed a test data set, and that was the data set that is shown here. So we saw that SVM model with sigmoid kernel was the best performing model for our case. And so we used that for the druggability prediction from so on and for comparison with existing methods. So this is the jackknife curve that is shown here, and the topmost one in pink that corresponds to the SVM model with sigmoid kernel. We titled it DRLiPS. It stands for Druggable RNA Ligand Binding Pocket Selector. So we have compared it with the 2 existing methods that I showed in the first introductory slide, and then 3 other methods that actually can predict binding sites. So one question that we asked ourselves was whether binding site prediction is equivalent to druggability prediction. Turns out it is not. When we did the predictions for this blind test data set, we saw that the methods are not able to identify really druggable binding sites. So the important learning aspect that we had in this particular study was that we found 6 structure-based descriptors that are actually very important for a druggable binding site in RNA. These were very interesting for us. So we found that polarity was very important. So number of hydrogen bond acceptors obviously was top of the list, followed by the number of cytosins in the binding site. And then 2 backbone torsion angles, actually epsilon zeta and alpha. Epsilon zeta, this is the difference between epsilon zeta torsion angles, and alpha is the individual torsion angle. So these actually occur sequentially if we look at the backbone, and they are all the 3 angles are actually very related to the flexibility of the RNA. So if there is a bend or unnatural orientation of the RNA, these angles are supposed to shoot up. So these angles coming up in our model development, they say that the flexibility is an important factor captured by the model. Also, the C2' endo puckering was captured as one of the important features, because often when the small molecule binds to the binding site, there is playing of the nucleotide happens. So the nucleotide like just flips out of the binding pocket, and that and sometimes they also directly interact with the ligand when they flip out, so that the volume of the binding site kind of increases and the interactions also become more favorable. So that kind of puckering is supposed to be important, and that we have captured in the model. And also molecular surface area was an important descriptor. So we did some Shapley and principal component analysis, which determine what is the order of feature importance, and I've given the order here. I also have the Sharp plots here. So in the Sharp plots, we have the feature value and the corresponding Shapley value. And this line in the middle, the 0, that shows us what is the separation between the highly druggable and less druggable binding sites, the positive and negative data set basically. And here we see that at least for the first 3 descriptors, there is a very clear separation between them when we look at the Shapley plots. But as the importance of the feature decreases, this discriminative ability also decreases. But nevertheless, as a combination, when they come together in a machine learning model, their importance comes up and they have a contribution to the prediction. So this is what we observed in this study. We also did a extensive analysis on model scope and limitations, whether the model can handle all special cases. So in this case, what we saw was that we picked an apo-hollow structure pairs. For example, like I said, apo and hollow structures can have widely varying conformational changes in RNA because it's highly dynamic. So we picked 7 apo-hollow structure pairs, mostly ribose switches from PDB. And here I've shown 2 examples, like adenine ribose switch and TPP ribose switch. So when adenine binds to adenine ribose switch, the local change in the conformation is minimal, but when TPP binds, the conformation is really different. So it goes from a completely open state to a completely closed state, and the backbone RMST from RNA align comes around 3.35 angstrom. So what we found is that when we looked at the Spearman's rank correlation between binding pocket rankings, the backbone RMST tolerance is 3 angstrom. So if the apo and hollow structures are different more than 3 angstrom in terms of the backbone RMST, the Spearman's correlation plummets down, and that means that the predictions might not be reliable after that point. So that is the tolerance criteria in case of comparing apo-hollow structure pair rankings from our model. In terms of mutations, so we took 10 different wild-type mutant structure pairs, and actually these mutations can be present anywhere in the RNA, and they have an effect on the KD value. So there are 4 categories of effects that we have given here. The KD can either decrease, this is with respect to the cognate ligand. The KD can either decrease, increase, it can have no effect, or it can have a loss of binding. And when we looked at the changes in the druggability score from the SVM model, in 8 out of 10 cases, this change in the KD is being captured by the model predictions. But in 2 cases, it's not able to capture, primarily because the mutations that are shown here, they are present outside the binding site. So for example, G19A in SAM1 ribose switch is far away from the binding site, but due to some allosteric effect, the KD is, there is an effect on the KD. So these cases cannot be captured by the model currently, since the features that we are computing are very specific only to the 6-point angstrom radius around the small molecule. So if a mutation comes outside that, the model is not able to capture that. We developed a web server again to host the model for public use. So here we have 2 prediction modes, basically known site mode and all sites mode. Known site mode meaning that the user has a site of interest in an RNA structure and they want to find out what is the druggability of it, whether it's druggable or not. So they give the PDB file. If it is an NMR model, NMR structure, you can tell us which model you're interested to analyze and what are the binding site residues or PDB ligand if it is bound in the structure. And we will pick it up for you and give the predictions. An example is shown here. This is a 16S rRNA, and here we have the A site of 16S rRNA, and here we have a molecule bound to it, and the 0.70 is the druggability score from the model. And if you, in all sites mode, actually, if you don't know the binding site, you just need to provide the structure, and the model will predict in the backend the binding sites for you, and it will give you a rank list for the complete structure. So that is the usage of the web server. And here for batch calculations, we have again provided GitHub code. Please check this out and use it in your studies if you're interested. And the last objective is that we wanted to develop, like I said, a basic end-to-end pipeline, an in silico pipeline for RNA-targeted drug discovery. So we have so far a database, we have a method to score interactions between RNA and small molecules, and we have a method to tell if a target is druggable or not. So with that, we wanted to build a basic pipeline. So for that, we took the standard, shape data set. The model organism that we chose was hepatitis C virus because there is a lot of structure-based data that is available experimentally. The data that we chose was the shape data set. Many of us here work with shape data set, so I'm not giving a elaborate introduction to shape. So shape stands for selective 2' hydroxyl acylation analyzed by primary extension. And shape gives you reactivity value, and this reactivity value gets converted into an information theory component called entropy using this formula. And low shape reactivity, low Shannon entropy, which I call here as type A structures. So these type A structures are ideal drug targets because they're highly base paired and they have a stable structure, and the conformational capture can happen basically. So their structure won't change that much. So we were focused on finding type A drug targets in hepatitis C virus genome. So we took the shape, derived a secondary structure, and we put it into this pipeline. So there are 4 modules in the pipeline. So first we start with target identification, and then structure prediction, target validation, and molecule screening. So we used RNA framework to analyze the shape data, and we also did covariance analysis to understand whether the base pairs that are there in the secondary structures have conservation evolutionarily across genotypes. So there are 7 genotypes in HCV, and for all the genotypes, we collected sequences and did this analysis, genotype specific as well as global genotype level conservation. That also we have analyzed. So we found 25 stable regions, 64% of them were conserved and 52% were novel. And then we did structure prediction using 7 different existing structure prediction programs. We gave the secondary structure from shape data as input to do this prediction. And we scored these models based on the, using qRNAs, we did a minimization and whether the minimized energy, that was one factor. And the second was whether it was able to preserve the experimental secondary structure provided based on the shape data. So those were our criteria, and we found that FAR2 performed best for this case, and we modeled all these 25 tertiary structures. And for target validation, we used the DRLiPS model that I was talking about in the last objective. We used that to predict binding sites, and the presence of druggable binding sites was considered as a validation for that target. And finally, for these binding sites, we did a massive high-throughput virtual screening. So we picked up like 31,000 compounds from various RNA-targeted libraries, including enamine, the CernaBio dataset, RBind, and etc. And then we did a massive virtual screening and identified some 24 potential molecules that can actually inhibit several regions in this HIV genomic elements. So this is our covariance analysis pipeline. Basically, it's a well-established pipeline used for several organisms in literature. So we built 3 iterations of covariance models using infernal, and we finally get the highly conserved structured genomic regions. And these are those genomic regions that we got in the HCV RNA genome. So we have 16 regions here. And for each region, wherever there is a green that is shown here, the green bar highlights that base pair is conserved evolutionarily in that genotype. So this region was very special, like the 762 to 816 region. So this region was very special because it was conserved in all the 7 genotypes. Although the base pairs that were conserved were different for each genotype, it was conserved in all the 7 HCV genotypes. So it can be a potential broad spectrum antiviral target. And we also found that structured regions in the 5' and 3' ends, the UTRs, were well conserved in comparison with those from the coding region. So that was another thing that we noticed. So these are the virtual screening results that I was talking about. So in this heat map, I have shown the, for the top 100 molecules, what is the distribution of the docking scores from RDoc. And this is the library-wise contribution to the top 100 molecules. So we see that after enamine is the most important contributor to these top 100 molecules. RSAM is also a second important contributor, followed by Robin and RBind. We couldn't find any other molecules from DRTL or the CernaBio pattern datasets in the top 100 molecules. These are the 24 promising inhibitors that we found. They have widely varying scaffolds, and these were actually identified with unique scaffold analysis compared to existing hepatitis C inhibitors. And here we see that this molecule is kind of special. I'm just giving a brief of what this molecule, why this molecule is special. So this molecule was found to interact with the E1, the envelope RNA, which is a coding region in the HCV genome. So this molecule has specific functional groups like phenylacetonitrile and piperazine, and these groups are also actually found in existing E1 protein inhibitors like chlorcyclazine or this piperazine benzonitrile derivative. These groups are preserved, but this was found for this RNA target, and the interactions were also really favorable. So we have stacking, we have hydrogen bonding with this with the various polar groups that are present here. The interactions were also really favorable. So this can be one of those compounds that can be assayed against both protein as well as RNA targets in HCV. But we would like to do further experimental validation to say more about these, but these are the compounds that we have shortlisted. So in conclusion, we have developed an array of resources for RNA-targeted drug discovery, and we have found several interesting sequence-based features, structure-based features, both global and binding site level, as well as small molecule descriptors that are very important for RNA small molecule interactions through machine learning. And we anticipate that these methods will be helpful and will lead to better therapeutic options. So in future, we would like to look at selectivity of RNA targets basically, because high binding affinity need not mean the molecules are highly selective. So we would like to look at selectivity, and which will help develop further method for looking at cross-target interactions in these molecules, which will help them progress further in pipelines. So these are some key references, and this is our group. I would like to sincerely thank my guide, Professor Michael Gromiha, for his support and motivation, and Dr. Nivedita for the opportunity to present our work in this esteemed forum. So we would be happy to take questions. Thank you.
Thanks, Somya, for the amazing presentation. So if anyone has a question, you can just paste it in the chat, or you can raise hand or ask, unmute yourself and ask. You can see a question. Somya, if you want to take the question from the chat.
Okay, yeah, and that's a great question. So actually, the length of the RNA, specific RNA secondary structures are definitely important, because like I said, the features that got chosen for the binding affinity case. So most of those features are very specific structure status descriptors, like the triplet descriptor for the mRNA I was talking about, or the the GCA pairs descriptors that I was talking about for aptamers. So specific RNA secondary structures, in the sense that these motifs, the presence of these, I would say, secondary structure motifs in this predicted secondary structure, those are quite important for the accuracy. And the length of the RNA, I'm not sure it plays a role here, because even though RNA is lengthy, we are still consolidating it into these 2000 descriptors. So when we consolidate the data, basically the length doesn't become a huge factor in the accuracy. Probably in the prediction time maybe, but not the accuracy, I think, because the server has to compute the features in the backend, and that might take time if the length of the RNA is long, like ribosome. So but the accuracy doesn't really get affected by the length, because we are converting it into a feature vector representation. Does that answer your question? Thank you.
Okay, if there are no other questions, so let us thank Professor Gromiha and Somya for the very interesting presentation. And yeah, I guess if anyone has a question, we still have time. You can raise hand or ask a question. Okay, so if there are no more questions, so thanks everyone for joining today's seminar, and we'll probably see you in 2 weeks. We'll have a seminar by Janusz on August 6th. So thank you, everyone.
Thank you for the opportunity.